Next is about antigen presenting cells and their interaction with T cells. Activation of both the humoral and cell mediated branches of the immune system require cytokines produced by Th cells. It is essential that activation of Th cells themselves be carefully regulated because a T cell response directed against self components can have fatal autoimmune consequences. One safeguard against unregulated activation of Th cells is that the antigen receptors of Th cells can recognize only antigen that is displayed together with class II MHC molecule on the surface of antigen presenting cells. These specialized cells, which include macrophages, B lymphocytes, and dendritic cells, are distinguished by two properties. They express class II MHC molecules on their membranes, and they can produce cytokines that cause Th cells to become activated. Antigen presenting cells first internalize antigen by either phagocytosis or endocytosis, and then display a part of that antigen on their membrane bound to a class II MHC molecule. The Th cell interacts with the antigen class II MHC molecule complex on the membranes of the antigen presenting cell. The antigen presenting cell then produces an additional signal leading to activation of the Th cell. Humoral and cellular immune response exhibit different effector functions. As mentioned earlier, immune response can be divided into humoral and cell-mediated response. Humoral immunity refers to immunity that can be conferred on a non-immune individual by administration of serum antibodies from an immune individual. In contrast, cell-mediated immunity can be transferred only by administration of T cells from an immune individual. The humoral branch of the immune system is at work in the interaction of B cells with antigen and their subsequent proliferation and differentiation into antibody-secreting plasma cells. Antibody functions as the effector of the humoral response by binding to antigen and facilitating its elimination. Antigen coated with antibody is eliminated in several ways. For example, antibody can cross-link several antigens, forming clusters that are more readily ingested by phagocytic cells. Binding of antibody to antigen on a microorganism can also activate the complement system, resulting in lysis of the foreign organisms. In addition, antibody can neutralize toxins or viral particles by coating them, which prevents them from binding to host cells. Effector T cells generated in response to antigen are responsible for cell-mediated immunity. Both activated Th cells and cytotoxic T lymphocytes serve as effector cells in cell-mediated immune reactions. Cytokines secreted by Th cells can activate various phagocytic cells, enabling them to phagocytose and kill microorganisms more effectively. This type of cell-mediated immune response is especially important in riding the host of cells and protozoa contained by infected host cells. CTLs participate in cell-mediated immune reactions by killing altered cells, including virus-infected cells and tumor cells. The antigen receptors of B and T lymphocytes are diverse. The antigenic specificity of each B cell is administered by the membrane-bound antigen-binding receptor, that is, the antibody. Then, the antigen receptors of B and T lymphocytes are diverse. The antigenic specificity of each B cell is determined by the membrane-bound antigen-binding receptor or antibody expressed by the cell. As a B cell matures in the bone marrow, its specificity is created by random rearrangements of a series of gene segments that encode the antibody molecule. At maturity, each B cell possesses a single functional gene encoding the antibody heavy chain and a single functional gene encoding the antibody light chain. All antibody molecules on a given B lymphocyte have identical specificity, giving each B lymphocyte and the clone of daughter cells to which it gives rise a distinct specificity for a single antigen. The mature B lymphocyte is therefore said to be antigenically committed. The random gene rearrangements during B cell maturation in the bone marrow generate an enormous number of different antigenic specificities. The resulting B cell population, which consists of individual B cells, each expressing A unique antibody is estimated to exhibit collectively more than 10 raised to 10 different antigenic specificities. A selection process in the bone marrow eliminates any B cells with membrane-bound antibody that recognizes self-components. This process is later discussed. The selection process helps to ensure that self-reactive antibodies or autoantibodies are not propagated. The attributes of specificity and diversity also characterize the antigen binding T cell receptor on T cells. As in B cell maturation, the process of T cell maturation includes random rearrangements of a series of gene segments that encode the cell's antigen binding receptor. Each T lymphocyte cell expresses about 10 raised to 5 receptors at which identical specificity for antigen. The random rearrangements of the TCR genes is capable of generating on the order of 10 raised to 9 unique antigenic specificities. The major histocompatibility complex molecules bind antigenic peptides. The major histocompatibility complex is a large genetic complex with multiple loci. This group of genes first gained attention as a barrier to tissue transplantation. Mismatched MHC genes lead to rejection of transplanted tissue and organs, hence the name histo-tissue compatibility. So loci within the MHC encode two major classes of membrane-bound glycoproteins, class 1 and class 2 MHC molecules. As noted above, TH cells generally recognize antigen combined with class 2 molecules, whereas TC cells generally recognize antigen combined with class 1 molecule. MHC molecules function as antigen recognition molecule, but they do not possess the fine specificity for antigen characteristic of antibodies and T cell receptors. Rather, each MHC molecule can bind to a spectrum of antigenic peptides, mostly derived from the degradation of protein molecules. In both class 1 and class 2 MHC molecule, there is a cleft within which the antigenic peptide sits and is presented to T lymphocytes. An antigen selection of lymphocytes causes clonal expansion. A mature immunocompetent animal contains a large number of antigen-reactive clones of T and B lymphocytes. The antigenic specificity of each of these clones is determined by the specificity of the antigen binding receptor on the membrane of the clone's lymphocytes. As noted above, the specificity of each T and B lymphocyte is determined before its contact with antigen by random gene rearrangements during maturation in the thymus or bone marrow. The role of antigen becomes critical when it interacts with the active, mature, antigenetically committed T and B lymphocytes, bringing about expansion of the population of cells with a given antigenic specificity. In this process of clonal selection, an antigen binds to a particular B or T cell and stimulates it to divide repeatedly into a clone of cells with the same antigenic specificity as the original parent cell. Clonal selection provides a framework for understanding the specificity and non-self recognition that is characteristic of adaptive immunity. Specificity is present because only lymphocytes whose receptors are specific for a given antigen will be clonally expanded and thus mobilized for an immune response. Self-non-self discrimination is accompanied by the elimination during development of lymphocytes bearing self-reactive receptors or the functional suppression of these cells if they reach maturity. Then immunologic memory is another consequence of clonal selection. During clonal selection, the number of lymphocytes specific for a given antigen is greatly amplified. Moreover, many of these lymphocytes, referred to as memory cells, have a longer lifespan than the naive lymphocytes from which they arise. The initial encounter of the naive immunocompetent lymphocyte with an antigen inducing a primary response, a later contact of the host with the antigen will induce a more rapid and heightened secondary response. The amplified population of memory cells accounts for the rapidity and intensity that distinguishes a secondary response from the primary response. In the humoral branch of immune system, antigen includes the clonal proliferation of B lymphocytes into antibody-secreting plasma cells and memory B cells. The primary response has a lag of approximately 5 to 7 days before antibody levels start to rise. This lag is the time required for activation of new B cells by antigen and TH cells and for the subsequent proliferation and differentiation of the activated B cells into plasma cells. Antibody levels peak in the primary response at about day 14 and then begin to drop off as the plasma cells begin to die. In the secondary response, a lag is much shorter. only 1 to 2 days. Antibody levels are much higher and they are sustained for much longer. The secondary response reflects the activity of the clonal expanded population of memory B cells. These memory cells respond to the antigen more rapidly than naive B cells. In addition, because there are many more memory cells than there were naive B cells for the primary response, more plasma cells are generated in the secondary response and antibody levels are consequently 100 to 1000-fold higher. In the cell-mediated branch of the immune system, the recognition of an antigen MHC complex by a specific mature T lymphocyte induces clonal proliferation into various T cells with effector functions, Th cells and CTLs, and into memory T cells. As with humoral immune response, secondary cell-mediated response are faster and stronger than primary. And immune dysfunction and its consequences. The above overview of innate and adaptive immunity depicts a multi-component interactive system that projects and protects the host from invasion by pathogens that cause infections, diseases, and form altered cells that can reproduce in an uncontrolled manner to cause cancer. This overview would not be complete without mentioning that the immune system can and does at times function improperly. Sometimes the immune system fails to protect the host adequately because of a deficiency, and sometimes it overreacts or misdirects its activities to cause discomfort, debilitating disease, or even death. There are several common manifestations of immune dysfunction: allergies and asthma, graft rejection and graft versus host disease, autoimmune diseases, immunodeficiency. Allergies and asthma are results of inappropriate immune response, often to common allergens such as plant pollen, food, or animal dander. The possibility that certain substances induced increased sensitivity or hypersensitivity rather than protection was recognized in about 1902 by Charles Riches, who attempted to immunize dogs against the toxins of a type of jellyfish, Physalia. He and his colleagues Paul Pocha observed that dogs exposed to sublethal doses of the toxin reacted almost instantly and fatally to subsequently challenge with minute amounts of the toxin, which it concluded that a successful immunization or vaccination results in phylaxis or protection, where the opposite result may occur, anaphylaxis, in which exposure to antigen can result in a potentially lethal sensitivity to the antigen if the exposure is repeated. And Richards received the Nobel Prize in 1913 for his discovery on the anaphylactic response. So fortunately, most allergic reactions in humans are not rapidly fatal. A specific allergic or anaphylactic response usually involves a type of antibody called IgE for immunoglobulin E. Binding of IgE to its specific antigen allergen releases substances that cause irritation and inflammation. When an allergic individual is exposed to an antigen, symptoms may include sneezing, wheezing, and difficulty in breathing, like asthma, dermatitis, or skin eruptions or hives, and in more extreme cases, strangulation due to blockage of airways by inflammation. A significant fraction of our health resources is expanded to care for those suffering from allergies and asthma. The frequency of allergy and asthma in the United States places these complaints among the most common reasons for a visit to the doctor's office or to the hostel emergency room. When the immune system encounters foreign cells or tissues, it responds strongly to rid the host of the invader. The same response may be raised against mutant host cells, including cancer cells. However, in some cases, the transplantation of cells or an organ from another individual, although viewed by the immune system as a foreign invasion, may be the only possible treatment for life-threatening disease. For example, it is estimated that more than 70,000 people in the United States alone would benefit from a kidney transplant. The fact that the immune system will attack and reject any transplanted organ that it recognizes as non-self raises a formidable barrier to this potentially life-saving treatment. An additional danger in transplantation is that any transplanted cell with immune function, for example, when bone marrow is transplanted to restore immune function, may view the new host as non-self and react against it. This reaction, which is termed graft versus host disease, can be fatal. The rejection reaction and graft versus host disease can be suppressed by drugs, but treatment with these drugs suppress general immune function so that the host is no longer protected by its own immune system and becomes susceptible to infectious disease. Transplantation studies have played a major role in the development of immunology. A Nobel Prize was awarded to Karl Landsteiner, mentioned above, for his contributions to the concept of immune specificity in 1930 for the discovery of human ABO blood groups, a finding that allowed blood transfusions to be carried out safely. In 1980s, G. Snell and some other scientists was recognized for discovery of the major histocompatibility complex, and in 1991, E. D. Thomas and Murray were awarded Nobel Prizes for advancements in transplantation immunity. Development of procedures that would allow a foreign organ to be accepted without suppressing immunity to all antigens remain a challenge for immunologists today. In certain individuals, the immune system malfunctions by losing its sense of self and non-self, which permits an immune attack on the host. This condition, autoimmunity, can cause a number of chronic debilitating disease. The symptoms of autoimmunity differ depending on which tissues and organs are under attack. For example, multiple sclerosis is due to an autoimmune attack on a protein in nerve sheaths on the brain and central nervous system. Crohn's disease is an attack on intestinal tissue, and rheumatoid arthritis is an attack on joints of the hand, feet, arms, and legs. The genetic and environmental factors that trigger and sustain autoimmune disease are very active areas of immunologic research, as in the search for improved treatments. If any components of innate or specific immunity are defective because of genetic abnormality, or if any immune function is lost because of damage by chemical, physical, or biological agents, the host suffers from immunodeficiency. The severity of the immunodeficiency disease depends on the number of affected components. A common type of immunodeficiency in North America is a selective immunodeficiency in which only one type of immunoglobulin, IgA, is lacking. The symptoms may be an increase in certain type of infections, or deficiency may even go unnoticed. In contrast, a rarer immunodeficiency called the severe combined immunodeficiency, shortly represented as SCID, SCID, which affects both B and T cells, may result in death from infection at an early age if untreated. Since the 1980s, the most common form of immunodeficiency has been acquired immunodeficiency syndrome, or AIDS, which results from infections with the retrovirus human immunodeficiency virus, or HIV. In AIDS, T helper cells are destroyed by HIV, causing a collapse of the immune system. It is estimated that 40 million people worldwide suffer from this disease, which, if not treated, is usually fatal within 8 to 10 years after infection. Although certain treatments can now prolong the life of AIDS patients, there is no known cure for this disease. This chapter, which we just learned with the help of two videos, I guess, one previous video and this one, has been a brief introduction to the immune system, and it has given a thumbnail stretch of how this complex system functions to protect the host from disease. The following chapters will examine the structure and function of the individual cells, organs, and molecules that make up the system. They will describe our current understanding of how the components of immunity interact and the experiments that allowed discovery of these mechanisms. Specific areas of applied immunology, such as immunity to infectious diseases, cancer, current vaccination practices, and the major types of immune dysfunction, are the base subjects for later chapters. Now let's learn about the topic called cells and organs of the immune system. In many cells, organs, and tissues of the immune system are found throughout the body. They can be classified functionally into two main groups. The primary lymphoid organs provide appropriate microenvironments for the development and maturation of lymphocytes. The secondary lymphoid organs trap antigen, generally from nearby tissues or vascular spaces and are sites where matured lymphocytes can interact effectively with antigen. Blood vessels or lymphatic systems connect these organs, uniting them into a functional whole. Carried within the blood and lymph and populating the lymphoid organs are various white blood cells or leukocytes that participate in the immune response. Of these cells, only the antigen-specific lymphocytes possesses the attributes of diversity, specificity, memory, and self-non-self recognition, the hallmarks of an adaptive immune response. Other leukocytes also play important roles, some as antigen-presenting cells and others participating as effector cells in the elimination of antigen, the phagocytosis or the secretion of immune effector molecules. Some leukocytes, especially T lymphocytes, secrete various protein molecules called cytokines. These molecules act as immunoregulatory hormones and play important roles in the coordination and regulation of immune responses. Here we describe the maturation of blood cells, the properties of various immune stem cells, and functions of the lymphoid organs. So, hematopoiesis. All blood cells arise from a type of cell called the hematopoietic stem cell, HSC. Stem cells are cells that can differentiate into other cell types. They are self-renewing, maintaining the population level by cell division. In humans, hematopoiesis, the formation and development of red and white blood cells begin in the embryonic yolk sac during the first weeks of development. Yolk sac stem cells differentiate into primitive erythroid cells that contain embryonic hemoglobin. By the third month of gestation, hematopoietic stem cells have migrated from the yolk sac to the fetal liver and subsequently colonized the spleen. These two organs have major roles in hematopoiesis from the third to the seventh month of gestation. After that, the differentiation of HSCs in the bone marrow become the major factor in hematopoiesis, and by birth there is little or no hematopoiesis in the liver and spleen. This migration of hematopoiesis during development form a series of early sites to bone marrow is very important. Hematopoiesis, we just learned or just mentioned, all blood cells arise from a type of cells called hematopoietic stem cells. Stem cells are cells that differentiate into other cell types. They are self-renewing, maintaining their population level by cell division. In humans, hematopoiesis, the formation and development of red and white blood cells begin in the embryonic yolk sac during the first weeks of development. Yolk sac stem cells differentiate into primitive erythroid cells that contain embryonic hemoglobin. By the third month of gestation, hematopoietic stem cells have migrated from the yolk sac to the fetal liver and subsequently colonize the spleen. These two organs have major roles in hematopoiesis from the third to the seventh months of gestation. After that, the differentiation of HCCs in the bone marrow become the major factor in hematopoiesis, and by birth there is little or no hematopoiesis in the liver and spleen. The migration of hematopoiesis during development from a series of early sites to bone marrow is very important. It is remarkable that every functionally specialized mature blood cell arises from an HSC. However, the study of hematopoietic stem cells is difficult for two reasons. They are scarce, normally fewer than one HSC per 5 into 10 raised to 4 cells in the bone marrow, and they are difficult to grow in vitro. As a result, the knowledge we have about how their proliferation and differentiation is regulated remains incomplete. By virtue of their capacity for self-renewal, hematopoietic stem cells are maintained at stable levels throughout adult life. However, when there is an increased demand for hematopoiesis, HSCs display an enormous proliferative capacity. This can be demonstrated in mice whose hematopoietic systems have been completely destroyed by a lethal dose of X-rays. Such irradiated mice die within 10 days unless they are infused. Then, irradiated mice die within 10 days unless they are infused with normal bone marrow cells from a syngenic, genetically identical mouse. Although a normal mouse has 3 into 10 raised to 8 bone marrow cells, infusion of only 10 raised to 4 to 10 raised to 5 bone marrow cells of the normal amount from a donor is sufficient to completely restore the hematopoietic system, which demonstrates the enormous capacity of HSCs for self-renewal. Early in hematopoiesis, a multipotent stem cell differentiates along one of two pathways, giving rise to either a lymphoid progenitor cell or a myeloid progenitor cell. Progenitor cells have lost the capacity for self-renewal and are committed to a particular cell lineage. Lymphoid progenitor cells give rise to B, T, and NK natural killer cells. Myeloid stem cells generate progenitors of red blood cells, erythrocytes. Many of the various white blood cells, neutrophils, eosinophils, basophils, monocytes, mast cells, dendritic cells, and platelet generating cells called megakaryocytes. In bone marrow, hematopoietic cells and their descendants grow, differentiate, and mature on a mesh-like scaffold of stromal cells, which include fat cells, endothelial cells, fibroblasts, and macrophages. Stromal cells influence the differentiation of hematopoietic stem cells by providing a hematopoietic induced microenvironment or HIM, consisting of a cellular matrix and factors that promote growth and differentiation. Many of these hematopoietic growth factors are soluble agents that arrive at their target cells by diffusion, whereas others are membrane-bound molecules on the surface of stromal cells that require cell-to-cell contact between the responding cells and the stromal cells. During hematopoiesis, erythrocytes and many different kinds of white blood cells descend from a few hematopoietic stem cells in a complicated process that involves many steps traversing a hierarchy of precursor populations. Why has such a complex process evolved to generate blood cells? Over a lifetime, a person will produce about 10 raised to 6 blood cells. This requires a great many cell divisions. John Dick has pointed out that cell division is error prone and provides an opportunity for the genome to suffer mutations, some of which may cause cancer. He has suggested that to make this potentially catastrophic event less likely, the blood forming system is organized as an ingenious hierarchy in which most of the proliferation takes place within more differentiated precursors rather than in the hematopoietic stem cell population itself. These progressively differentiating precursors are not self-renewing, and the mature blood cells they form are either incapable of division or only divide under special circumstances. So consequently, the chance of generating cancer in HSCs and their immediate descendants is lower, even if it is not reduced to zero. Hematopoiesis is regulated at the genetic level. The development of pluripotent hematopoietic stem cells into different cell types require the expression of different sets of lineage determining and lineage specific genes at appropriate times and in the correct order. The proteins specified by the genes are critical components of regulatory networks that direct the differentiation of the stem cell and its descendants. Much of what we know about the dependence of hematopoiesis on a particular gene comes from studies of mice in which a gene has been inactivated or knocked out by targeted disruption, which blocks the production of the protein that it encodes. If mice fail to produce red cells or particular white blood cells when a gene is knocked out, we conclude that the protein specified by the gene is necessary for development of those cells. Knockout technology is one of the most powerful tools available for determining the roles of particular genes in a broad range of processes, and it has made important contributions to the identification of many genes that regulate hematopoiesis. Although much remains to be learned, targeted disruption and other approaches have identified a number of transcription factors that play important roles in hematopoiesis. Some of these transcription factors affect many different hematopoietic lineages, and others affect only a single lineage, such as the development pathway that leads to lymphocytes. One transcription factor that affects multiple lineages is TATA2, a member of family of transcription factors that recognize the tetranucleotide sequence TATA, a motif found in target genes. A functional TATA2 gene, which specifies this transcription factor, is essential for the development of the lymphoid, erythroid, and myeloid lineages, as might be expected. animals in which this gene is disrupted die during embryonic development. In contrast to GATA2, another transcription factor, Icaros, is required only for the development of cells of the lymphoid lineage. Although Icaros knockout mice do not produce significant numbers of B, T, and NK cells, their production of erythrocytes, granulocytes, and other cells of the myeloid lineage is unimpaired. Icaros knockout mice survive embryonic development, but they are severely compromised immunologically and die of infections at an early age. Yet another transcription regulator, BMI1, is a transcription repressor that is a key determinant of the ability of HCS to self-renew. When the gene for B1, which is BMI1, which is highly expressed in HSCs of humans and mice, is knocked out, and BMI1 deficient mice die within 2 months of birth. The cause of death is the eventual failure of the bone marrow to generate red and white blood cells. This bone marrow failure is traced to a lack of self-renewal by HSCs. Then, next topic, hematopoietic hemostasis involves many factors, which are the hematopoiesis is a steady state process in which mature blood cells are produced at the same rate at which they are lost. The principal cause of blood cell loss is aging. The average erythrocyte has a lifespan of 120 days before it is phagocytosed and digested by macrophages in the spleen. The various white blood cells have lifespans ranging from a day for neutrophils as long as 20 to 30 years for some T lymphocytes. To maintain steady state levels, the average human being must produce an estimated 3.7 into 10 raised to 11 white blood cells per day. This massive system is regulated by complex mechanisms that affect all of the individual cell types, and ultimately the number of cells in any hematopoietic lineage is set by a balance between the number of cells removed by cell death and the number that arise from division and differentiation. Any one or a combination of regulatory factors can affect rates of cell reproduction and differentiation. These factors can also determine whether a hematopoietic cell is induced to die. Programmed cell death is an essential homeostatic mechanism. Programmed cell death, an induced and ordered process in which the cell activity participates in bringing about its own demise, is a critical factor in the homeostatic regulation of many types of cell populations, including those of the hematopoietic system. Cells undergoing programmed cell death often exhibit distinctive morphological changes, collectively referred to as apoptosis. These changes include a pronounced decrease in cell volume, modification of the cytoskeleton, which results in membrane blubbing, a condensation of chromatin, and degradation of DNA into fragments. Following these morphologic changes, an apoptotic cell sheds tiny membrane-bound apoptotic bodies containing intact organelles. Macrophages phagocytose apoptotic bodies and cells in the advanced stages of apoptosis. This ensures that their intracellular contents, including proteolytic and other lytic enzymes, cationic proteins, and oxidizing molecules, are not released into surrounding tissue. As a consequence, apoptosis does not induce a local inflammatory response. Apoptosis differs markedly from necrosis, a change associated with cell death rising from injury. In necrosis, injured cells swell and burst, releasing their contents and possibly triggering a damaging inflammatory response. Each of the leukocytes produced by hematopoiesis has a characteristic lifespan and then dies by programmed cell death. In the adult human, for example, there are about 5 into 10 raised to 10 neutrophils in the circulation. These cells have a lifespan of only a day before programmed cell death is initiated. A stable number of these cells is maintained by constant neutrophil production. Programmed cell death also plays a role in maintaining proper number of hematopoietic progenitor cells for erythrocytes and various types of leukocytes. Beyond hematopoiesis, apoptosis is important in such immunological processes as tolerance and the killing of target cells by cytotoxic T cells or natural killer cells. Details of the mechanisms underlying apoptosis are very important. And the expression of several genes accompanies apoptosis in leukocytes and other cell types. Some of the proteins specified by these genes include induce apoptosis, others are critical as apoptosis progresses and still others inhibit apoptosis. For example, apoptosis can be induced in lymphocytes by radiation, but only if the protein p53 is present. Many cell deaths are induced by signals from Fas, a molecule present on the surface of many cells. And proteases known as caspases take part in a cascade of reactions that lead to apoptosis. On the other hand, members of the BCL2 B-cell lymphoma 2 family of genes, BCL2 and BCL-XL, encode protein products that inhibit apoptosis. Interestingly, a first member of this gene family to be discovered, BCL2, was found in studies that were concerned not with cell death but with the uncontrolled proliferation of B cells in a type of cancer known as a B cell lymphoma. In this case, the BCL2 gene was near the breakpoint of a chromosomal translocation in a human B cell lymphoma. The translocation moved the BCL2 gene into the immunoglobulin heavy chain locus, resulting in transcription of BCL2 along with the immunoglobulin gene, with consequent overproduction of the encoded BCL2 protein by the lymphoma cells. The resulting high levels of BCL2 are thought to help transform lymphoid cells into cancerous lymphoma cells by inhibiting the signals that would normally induce apoptotic cell death. BCL2 levels have been found to play an important role in regulating the normal lifespan of various hematopoietic cell lineages, including lymphocytes. A normal adult has about 5 liters of blood with about 2000 lymphocytes per millimeter cube for a total of about 10 raised to 11 circulating lymphocytes. During acute infection, the lymphocyte count increases fourfold or more, producing a circulating lymphocyte level of over 4 into 10 raised to 11. The immune system cannot sustain such a massive increase in cell numbers for an extended period, so the system must eliminate unneeded activated lymphocytes once the antigenic threat has passed. Activated lymphocytes have been found to express lower levels of BCL2 and therefore are more susceptible to the induction of apoptotic death than are naive lymphocytes or memory cells. However, if the lymphocytes continue to be activated by antigen, signals received during activation block the apoptotic signal. As antigen levels subside, the levels of the signals that block apoptosis diminishes and the lymphocytes begin to die by apoptosis, and hematopoietic stem cells can be enriched. Eve Weissman and colleagues developed a novel way of enriching the concentration of mouse hematopoietic stem cells, which normally constitute less than 0.05% of all bone marrow cells in mice. Their approach relies on the use of antibodies specific for molecules known as differentiation antigens, which are expressed only by particular cell types. They exposed bone marrow samples to antibodies that had been labeled with a fluorescent compound and were specific for the differentiation antigens expressed on the surface of mature red and white blood cells, but not on stem cells. The labeled cells were then removed by flow cytometry with a fluorescence activated cell sorted. After each sorting, the remaining cells were assayed to determine the minimum number needed for restoration of hematopoiesis in a lethally X-irradiated mouse. As the pluripotent stem cells become relatively more numerous in the remaining population, fewer and fewer cells were needed to restore hematopoiesis in the system. Removal of the hematopoietic cells by selecting for the presence of differentiation antigens allowed a 50 to 200 fold enrichment of pluripotent stem cells. To further enrich the pluripotent stem cells, the remaining cells were incubated with various antibodies raised against the cells likely to be in the early stages of hematopoiesis. One of these antibodies recognized a differentiation antigen called stem cell antigen 1. Treatment with this antibody aided capture of undifferentiated stem cells and yielded a preparation so enriched in pluripotent stem cells that an aliquot containing only 30 to 100 cells routinely restored hematopoiesis in a lethally X-irradiated mouse, whereas more than 10 days to 4 non-enriched bone marrow cells were needed for restoration. Using a refinement of this approach, H. Nakoshi and his colleagues have devised a procedure so effective that in 1 out of 5 lethally irradiated mice, a single hematopoietic cell can restore both myeloid and lymphoid lineages. It has been found that CD34, a marker found on about 1% of hematopoietic cells, although not unique to stem cells, is found on a small population of cells that includes stem cells. The administration of human cell population suitably enriched for CD34 plus cells. Plus in the sense that plus indicates that the factor is present on the cell membrane. Okay, can constitute a patient's entire hematopoietic system. A major tool in studies to identify and characterize the human hematopoietic stem cell is the use of SCID, or severe combined immunodeficiency mice as an in vivo assay system for the presence and function of HSCs. SCID mice do not have B and T lymphocytes and are unable to mount adaptive immune response such as those that act in the normal rejection of foreign cells, tissues, and organs. Consequently, these animals do not reject transplanted human cell populations containing HSCs or tissues such as thymus and bone marrow. Immunodeficient mice have been useful surrogates or alternative hosts for in vitro studies of human stem cells. Implanted with fragments of human thymus and bone marrow, SCID mice support the differentiation of human hematopoietic stem cells into mature hematopoietic cells. The system has made possible the study of subpopulations of CD34 plus cells and the effect of human growth factors on the differentiation of various hematopoietic lineages. And cells of the immune system, lymphocytes bearing antigen receptors are the central cells of adaptive immunity and are responsible for its signature properties of diversity, specificity, and memory. Important as lymphocytes are, other types of white blood cells also play essential roles in adaptive immunity, presenting antigens, secreting cytokines, and engulfing and destroying microorganisms. Furthermore, as we will see in next chapter, the innate immune system, which shares many cells with the adaptive immune system, plays an indispensable collaborative role in the introduction of adaptive responses. Cells of the immune system. We just mentioned about this. Then lymphoid cells. We just mentioned about lymphoid cells. Lymphocytes constitute 20% to 40% of the body's white blood cells and 99% of cells in the lymph. There are approximately a trillion, 10 raised to 12 lymphocytes in the human body. Lymphocytes circulate continuously in the blood and lymph and are capable of migrating into the tissue spaces and lymphoid organs, serving thereby as a bridge between parts of the immune system. The lymphocytes can be broadly classified into three major populations: B cells, T cells, and natural killer cells. On the basis of function and cell membrane components, key cells of adaptive immunity, B cells and T cells, each bear their own distinctive family of antigen receptors. Natural killer cells are large granular lymphocytes. Granular refers to their grainy appearance under a microscope and are part of the innate immune system and do not express the set of surface markers that characterize B or T cells. B and T lymphocytes that have not interacted with antigen, referred to as naive or unprimed, are small, motile, non-phagocytic cells that cannot be distinguished from each other morphologically. In their inactive state, they remain in the G0 phase of the cell cycle, known as small lymphocytes. These cells are only about 6 μm in diameter. Their cytoplasm forms a barely discernible rim around the nucleus. Small lymphocytes have densely packed chromatin, few mitochondria, and a poorly developed endoplasmic reticulum and Golgi apparatus. The naive lymphocyte is generally thought to have a short lifespan. Under appropriate conditions, the interaction of small lymphocytes with antigen induces these cells to progress through the cell cycle from G0 to G1 and subsequently from S and G2 and M. As the cell cycle proceeds, lymphocytes enlarge into 1.5 μm diameter cells called lymphoblasts. These cells have a higher cytoplasm to nucleus ratio and more organelle complexity than small lymphocytes. So lymphoblasts proliferate and eventually differentiate into effector cells or into memory cells. Effector cells function in various ways to eliminate antigen. These cells have lifespans generally ranging from few days to a few weeks. Natural killer cells are large granular lymphocytes. Granular refers to their grainy appearance under the microscope that are part of the innate immune system and do not express the set of surface markers that characterize B or T cells. B and T lymphocytes that have not interacted with antigen to a snail or unprimed are small, motile, non-phagocytic cells that cannot be distinguished from each other morphologically. In their inactive state, they remain in the G0 phase of the cell cycle. Known as small lymphocytes, these cells are only about 6 μm in diameter. Their cytoplasm forms a barely discernible rim around the nucleus. Small lymphocytes have densely packed chromatin, few mitochondria, and a poorly developed endoplasmic reticulum and Golgi apparatus. The naive lymphocyte is generally thought to have a short lifespan. Under appropriate conditions, the interaction of small lymphocytes with antigen induces these cells to progress through the cell cycle from G0 to G1 and subsequently into S, G2 and M. As the cell cycle proceeds, lymphocytes enlarge into 1.5 μm diameter cells called lymphoblasts. These cells have a higher cytoplasm to nucleus ratio and more organellar complexity than small lymphocytes. Lymphoblasts proliferate and eventually differentiate into effector cells or into memory cells. Effector cells function in various ways to eliminate antigen. These cells have lifespans generally ranging from a few days to a few weeks. Plasma cells, the antibody secreting effector cells of the B cell lineage, have a characteristic cytoplasm that contains abundant concentric layers of endoplasmic reticulum to support their high rate of protein synthesis and many Golgi vesicles. The effector cells of the T cell lineage include a cytokine secreting T helper cell, Th cell, and antigen activating mature cells of the cytotoxic lymphocyte, TC cells, lineage known as CTLs, cytotoxic T lymphocytes. Some of the progeny of B and T lymphoblasts differentiate into memory cells. The persistence of this population of cells is responsible for lifelong immunity to many pathogens. Memory cells look like small lymphocytes but can be distinguished from new cells by the presence or absence of certain molecules on their cell membranes. Different lineages or maturational stages or lymphocytes can be distinguished by their expression of membrane molecules recognized by particular monoclonal antibodies that are specific for a single epitope of an antigen. All of the monoclonal antibodies that tracked with a particular membrane molecule are grouped together as a cluster of differentiation. Each new monoclonal antibody that recognizes a leukocyte membrane molecule is analyzed to determine if it falls within a recognized CD designation. If it does not, it is given a new CD designation reflecting a new membrane molecule. Although the CD nomenclature was originally developed for the membrane molecules of human leukocytes, the homologous membrane molecules of other species, such as mice, are commonly referred to by the same CD designations. Some common CD molecules, often referred to as CD markers, found on human lymphocytes. However, this is only a partial listing of the approximately 250 CD markers that have been described. A complete list and description of non-CD markers is very important. The general characteristics of functions of B and T lymphocytes are reviewed briefly in the next sections. These central cells of the immune system will be examined much more fully in later chapters. Next, B lymphocytes, B cells. B cells. The B lymphocytes, also known as B cells, derived its later designation from its site of maturation in the bursa of Fabricius in birds, and name turned out to be apt as bone marrow. Is its major site of maturation in a number of mammalian species, including humans and mice. Mature B cells are definitely distinguished from other lymphocytes and all other cells by their synthesis and display of membrane-bound immunoglobulin antibody molecules which serve as receptors for antigen. Each of the approximately 1.5 into 10 raised to 5 molecules of antibody on the membrane of a single B cell has an identical binding site for antigen when a new B cell, one that has not previously encountered antigen, first encounters the antigen that matches its membrane-bound antibody. The binding of the antigen to the antibody causes the cell to divide rapidly. Its progeny differentiate into effector cells called plasma cells and into memory B cells. Memory B cells have a longer lifespan than naive cells and they express the same membrane-bound antibody as their parent B cell. Plasma cells, on the other hand, produce the antibody in a form that can be secreted and have little or no membrane-bound antibody. Plasma cells are end-stage cells and do not divide. Although some long-lived populations can be found in bone marrow, many die within 1 to 2 weeks. They are highly specialized for secretion of antibody, and a single cell is estimated to be capable of secreting from a few hundred to more than 1,000 molecules of antibody per second. In T lymphocytes or T cells, T lymphocytes derive their later designation from their site of maturation on the thymus. During its maturation within the thymus, the T cell comes to express on its membrane a unique antigen binding molecule called the T cell receptor. Unlike membrane bound antibodies on B cells, which can recognize antigen alone, T cell receptors only recognize antigen that is bound to cell membrane protein called major histocompatibility complex molecules. MHC molecules that function in this recognition event, which is termed antigen presentation, are genetically diverse polymorphic glycoproteins found on cell membranes. There are two major types of MHC molecules, class 1 MHC molecules expressed by nearly all nucleated cells of vertebrate species, and class 2 MHC molecules which are expressed by only a few cell types that are specialized for antigen presentation. When a T cell recognizes antigen combined with an MHC molecule on a cell under appropriate circumstances, the T cell proliferates and differentiates into various effector T cells and memory T cells. There are two well-defined subpopulations of T cells, T helper cells, TH cells, and T cytotoxic T cells, TC cells. Recently, a third cell population, T regulatory cells, has been characterized. The T helper and T cytotoxic cells can be distinguished from one another by the presence of either CD4 or CD8 membrane glycoproteins on their surface. T cell displaying CD4 generally function as TH cells, whereas those displaying CD8 generally function as TC cells. Thus, the ratio of TH to TC cells in a simple sample can be approximated by assuming the number of CD4 plus and CD8 plus T cells. This ratio is approximately 2:1 in normal human peripheral blood, but it may be significantly altered by immunodeficiency disease, autoimmune disease, and other disorders. Following activation by interaction with appropriate antigen MHC complexes, TH cells differentiate into effector cells that enable or help the activation of T cells, TC cells, macrophages, and various other cells that participate in the immune response. Alternatively, some TH cells differentiate into memory cells instead of effector cells. Recognition of antigen MHC complexes by a TC cell triggers its proliferation and differentiation into an effector cell called a cytotoxic T lymphocyte, CTL, or into a memory cell. The CTL has a vital function in monitoring the cells of the body and eliminating any that display foreign antigen complexed with class 1 MHC, such as virus infected cells, tumor cells, and cells of a foreign tissue graft. T regulatory cells are identified by the presence of both CD4 and CD25 on their membranes. However, unlike CD4 bearing T helper sites, T reg in the sense regulatory cells suppresses immune responses. They are negative regulators of immune system like T, H, and TC cells. Members of the T reg subpopulation of T cells may be progenitors of memory cells. Then B and T populations arise subpopulations of clones. All of the antigen receptors on a given B or T cell have identical structures and therefore are identical specificities for antigen. If a given lymphocyte divides to form two daughter cells, both the daughters bear antigen receptors with antigen specificities identical to each other and to the parental cell from which they arose, and so will any descendants they produce. The resulting population of lymphocytes, all arising from the same founding lymphocytes, is a clone. At any given moment, a human or a mouse will contain tens of thousands, perhaps 100,000 distinct T and B cell clones. Each clone distinguished by its own distinctive and identical cohort of antigen receptors. Contact with antigen includes those cells to proliferate and differentiate. The products of this process include both effector cells and memory cells. The effector cells carry out specific functions while the memory cells persist in the host and on re-challenge with some antigen mediate a response that is both quicker and greater magnitude. The first encounter is termed a primary response and the re-encounter a secondary response. Natural killer cells. The body contains a small population of large granular lymphocytes called natural killer cells that display cytotoxic activity against a wide range of tumor cells and against cells infected with some, but not all viruses. An extraordinary feature of these cells, which constitute 50% to 10% of lymphocytes in human peripheral blood, is their ability to recognize tumor or viral infected cells despite lacking antigen-specific receptors. Natural killer cells are part of the innate immune system, and most do not have T cell receptors or immunoglobulin incorporated in their plasma membranes. In other words, they do not express the membrane molecules and receptors that distinguish B and T lineages. NK cells recognize potential target cells in two different ways. In some cases, an NK cell employs NK cell receptors to distinguish abnormalities such as reduction in the display of class 1 MHC molecule or the unused profile of surface antigens displayed by some tumor cells and cells infected by some virus. In addition, some tumor cells and cells infected by certain viruses display antigens against which the immune system has raised an antibody response, so that anti-tumor or antiviral antibodies are bound to the surface. Because NK cells express a membrane receptor CD16 for a specific region of the antibody molecule, they can attach to these antibodies and subsequently destroy the targeted cells. This is an example of a process known as antibody-dependent cell-mediated cytotoxicity, ADCC. The exact mechanism of NK cell cytotoxicity, the focus of much current experimental study, is very important. Then recognition has recently been growing of another cell type, the NKT cell, which has some of the characteristics of both T cells and NK cells. Like T cells, NKT cells have a T cell receptor, TCRs. Unlike most T cells, the TCRs of NKT cells interact with MHC-like molecules called CD1 rather than with class 1 or class 2 MHC molecules like NK cells. They have variable levels of CD16 and other receptors typical of NK cells, and they can kill target cells. A population of activated NKT cells can rapidly secrete large amounts of cytokines needed to support antibody production by B cells, as well as inflammation and the development and expansion of cytotoxic T cells. So, determining the exact roles played by NKT cells in immunity is of great interest. Then, a quick recap about mononuclear phagocytes. The mononuclear phagocytic system consists of monocytes circulating in the blood and macrophages in the tissue. During hematopoiesis in the bone marrow, granulocyte monocyte progenitor cells differentiate into promonocytes, which leave the bone marrow and enter the blood, where they further differentiate into mature monocytes. Monocytes circulate in the bloodstream for about 8 hours, during which time they enlarge. They then migrate into the tissues and differentiate into specific tissue macrophages. So, differentiation of a monocyte into a tissue macrophage involves a number of changes. The cell enlarges 5 to 10 fold. Its intracellular organelles increase in both number and complexity, and it acquires increased phagocytic ability, produces higher levels of hydrolytic enzymes, and begins to secrete a variety of soluble factors. Macrophages are dispersed throughout the body. Some take up residence in particular tissue, becoming fixed macrophages, whereas others remain motile and are called free or wandering macrophages. Free macrophages travel by amyloid movement throughout the tissue. Macrophages, like cells, serve different functions in their tissues and are named according to their tissue location, like intestinal macrophages in the gut, alveolar macrophages in the lung, then histiocytes in connective tissue, Kupffer cells in the liver, then mesangial cells in the kidney, microglial cells in the brain, Then osteoclasts in bone. Macrophages are activated by a variety of stimuli in the course of an immune response. Phagocytosis of particulate antigens or contact with the receptors that sense molecules present in